Good afternoon, students. Hope you all are fine during this lockdown period. So, in our previous class, we have discussed about principles of genetics and a brief recap of what we have studied. In Mendelism, we have studied the law of dominance, segregation, and independent assortment. We have discussed few crosses like monohybrid, dihybrid, test, and backcross. We have also discussed about homozygous and heterozygous allele. Further, we have discussed about crosses which do not follow Mendelian inheritance. A brief recap, you must have remembered that during the class I taught you that Mendel performed all the experiments using the pea plant. Years after Mendel's discovery, since he was unable to predict why such phenomena are occurring, we have came across the discovery of DNA, the chromosomes and whatnot. In due course of time, in nature, we have found that there are certain examples which deviate from Mendel's law of inheritance. In your book, the Veerbala Rastogi, you will find that there are examples of incomplete dominance, codominance as in our blood group, epistasis, complementary gene, duplicate multiple genes, polygenes and pleiotropism. To have a brief recap of all these topics, please go through your book and you will find what we have studied. So, in today's topic, we will discuss about crossing over, recombination, and linkage. Now, as we all know, crossing over takes place in the meiosis stage. I hope you remember about the difference between meiosis and mitosis. A brief recap, meiosis usually takes place only in the germ cells. Now, meiosis, as we know, has four stages: prophase 1, metaphase 1, anaphase 1, and telophase 1, followed by meiosis 2, which is prophase 2, metaphase 2, anaphase 2, and telophase 2. The prophase 1 is the most interesting part of meiosis 1, where in the Pachytene stage the crossing over takes place. If you look at the image towards the left hand side, where it is shown that how two homologous chromosomes combines and results in crossing over during the Pachytene stage. Let's have a look towards the image at the right hand side of the slide. Two homologous chromosomes, one labeled in blue color and the other one labeled in the red color. After the crossing over takes place between two non-sister chromatids of the homologous chromosome, we can see the exchange of gene between the two chromosomes. So let's have a look. In the blue colored chromosome, we have three genes, capital lettered A, B and C. And in the red color chromosome, we have three genes, small letter A, B and C. Now these two genes are basically the alleles of each other, as we have discussed in the class previously. Now once the crossing over takes place between the non-sister chromatids of the homologous chromosome, we get the recombinant products as capital A, capital V and small c, followed by small a, small v and capital C. So if you have a closer look towards the diagram in the right hand side, the lower part, so see, in one chromatid of the blue colored chromosome, the chromosomes have the same set of gene pairs, capital A, B and C. But on the other chromatid of the same chromosome, we have the recombinant product, A, B and small c. The same thing has occurred in the red chromosome also. Now let me tell you something which you will come across during the later part of the slides, that crossing over can occur one point, as you can see in this chromosome. It can also occur in 2 points, or it can also occur in 3 points, as we will see in the next slide. So the image 1 of this slide shows the previous diagram which we have discussed in the previous slide. As you can see, the blue colored chromosome and the pink colored chromosome, both are homologous pair. They have the gene pair as capital A, capital B and small b. Both of them are allele of each other. Crossing over took place and which resulted in the formation of recombinants as well as non-recombinants. So see the recombinants part, we have capital A and small b and small a and capital B, and the non-recombinants part have capital A, capital B and small b, similar to the parent chromosomes. Now why they have mentioned gametes? Because all the four chromatids of the individual homologous chromosome have separated. So initially it was 2n and right now it is n, that is haploid. Coming to image 2, so have a look at this image very carefully. So see, two diagrams are shown here. One where no crossing over took place. In the red and blue color chromosome, the alleles do not cross over with each other. So red color chromosome has capital A and capital B, and the blue colored one has small a and small b. Even after meiosis II, when the four gametes are formed, they do not cross over with each other. These kind of scenario can take place during certain times of cell division. So we have learned that it is not mandatory that every set of meiosis will result in a crossing over product, right? And if the crossing over takes place, we will see the similar phenomenon which we have discussed on the image 1, that the red color chromosome and the non-sister chromatids of the red and blue colored chromosome cross over with each other and resulted in recombinants and non-recombinants. Now non-recombinants have the similar kind of gene set as that of the parent chromosome, like the chromosome red colored has capital A and capital B, and the blue colored chromosome has small a and small b, similar to the parent chromosome. But in the recombinant one, we can see the capital A combines with small b and small a combines with capital B. Now what is actually the result of this crossing over? As during the class I have mentioned several times that we as individuals are different. We are different from our parents, although we are the product of our parent cells, that is, the sperm cell of the father and the ova cell of the mother has combined together to give rise to a diploid zygote. But still we are different. We have something called individuality, and this individuality has came as a product of crossing over which has taken place in the individual parents. So I hope we are now clear on the grounds that what is crossing over and what is the result of crossing over, that is recombination. And as a recombination we create recombinants as well as non-recombinants. Now the most important part of this chapter, linkage. Now what is linkage? If we talk in the general terms of English vocabulary, linkage is something that means bonded together, or we are we will not segregate individually, we will get bonded together. Okay, so in chromosome also, crossing over usually takes place between two non-sister chromatids. Now think about crossing over. It is basically crossing between two non-sister chromatids at a particular distance, right? Now think about the image which is shown towards the left hand side of the slide. Two genes, gene 1 and 2, they are so close to each other that they cannot get segregated, or it will be better to say that even if the crossing over take place between the chromosomes having the gene orientation similar to gene 1 and 2, the crossing over products cannot migrate away from each other. Just have a look at the chromosomes shown in the middle of the slide. So gene 1 and gene 2 are located on two different chromosomes. So they can easily cross over between each other and hence they are labeled as not linked. The third chromosome, where the gene 1 and 2 are situated at a huge gap between to each other, even here also the recombinants will be developed and the crossing over point will be easily created. To clear our perception about linkage and non-linkage genes, let's have a look at this diagram. So as I said that if the gene orientation or the chromosome segregation follows Mendelian genetics, they will assort independently. Okay, according to the Mendel's third law of genetics. Now, in independent assortment, as we can see, the two chromosomes, one labeled red and the other labeled green, capital A and capital B, followed by the alleles small a and small b, they perform a crossing over and recombinants take place. As you can see, the small a capital B and capital A small b. But, as I said, for crossing over to take place and the genes to segregate independently, the gene has to be at a particular distance. If you see at the another image where the heading is labeled as linkage, just look at the distance between two genes. Gene on red colored chromosome and gene on green colored chromosome, capital A, capital B and small b, they are so close to each other that even if crossing over takes place, the crossing over product do not give rise to the recombinants. The main reason for not giving rise to recombinants is the genetic distance, which is known as the map distance, which we will see later on. And map distance is basically the distance between two gene pairs. Okay? So here are certain images how the genetic map actually looks like. So we will discuss about the genetic map in detail and how we map the genes. Now, you must have done the topo sheet mapping in geography. Now, what we actually do, mapping means pointing a specific location on the entire map. Now, think about genes located on chromosomes, which are nothing but a form of DNA. Now, the DNA is so small in size that we simply cannot have a look at it. Right? You cannot hold up a DNA in the on your hand, which is present inside your cell. So how actually are we targeting to map the genes? Do we have a specific scale for that? Well, yes, we do have. Now that scale is known as recombination frequency. Now what is actually recombination frequency? So we will discuss that in next slide. So initially, to understand linkage, you need to get around some terms, right? Now linkage definition has already been discussed to you. The another definition is of recombination frequency, which is the number of recombinants by the total number of progenies. The another thing which you need to know is centimorgan. Now what is centimorgan? Centimorgan is actually the unit of genetic map, distance. Just like when you measure distance in physics, you measure it in SI unit and that is by centimeter or millimeter or kilometer. Similarly, in genetic map distance measurement also, the unit is something called centimorgan. Now, how the term centimorgan came up? It's a brief story between Morgan, who was the scientist, and his student by the name Stuart Went. So it took Stuart Went one night to decide how can we map the genes between which are located on particular location of a chromosome. And because of that, Morgan and his student Stuart Went, both of them received Nobel Prize. So in this slide, we are having a closer look about how genes are mapped on a chromosome and how actually the cytogenetic map looks like. In the next slide, we will discuss how to find the genetic map. So in diagram A, we are talking about three genes A, B and C. Now have a look. The distance between A and B is 13.2 centimorgan, small c and capital M. The distance between B and C is 6.4 centimorgan and the total distance between A and C is 18.5 centimorgan. Okay, so how this particular digits came up? So see, if you have a look at image B, where the genetic distance between the three genes are shown and the recombination frequency between A and B, that means these two genes A and B are located on two chromatids of a homologous chromosome. Now the number of times the chromosome undergo crossing over, the resulting gene pairs between the crossing over product between gene A and B is 13.2%. That means out of 100%, only 13.2% are the chances that gene A and B will cross with each other, and the rest of the time they won't cross. Now see, since gene B and C are very close to each other, the recombination frequency is very less. And since A and C are very far away from each other, the recombination frequency is very high. Now, if you all remember what I said during the explanation of linkage, that the far the genes are, the more are the chances of crossing over. The less far the genes are, the lesser are the chances of crossing over. And that thing is totally revealed in image B. Since gene A and B are very close, are far away from each other, the recombination frequency is 13.2, and since gene B and C are less far from each other, the recombination frequency is less. That means the genes segregate together, they do not segregate individually, and hence from this diagram we can say that gene B and C are more linked to each other than gene A and B. Similarly, we say that gene A and C are less linked to each other in comparison to gene A and B and gene pair B and C. In diagram C, as we can see, the recombination frequency is calculated by total number of recombinants by total number of progeny into 100. Okay, go through your book, and I hope since this is the first class on this linkage, we will discuss about this in detail once the lockdown is over. In the meantime, go through Veer Bala Rastogi as well as your fundamentals of biology of Willy publication to have a brief idea what linkage actually is. Enjoyed the class? Thank you.